Thank you to Terrapin and the organizer for the invitation to present on this topic. Claudia presented on PTC and their efforts with Adalurin and DMD. And in many ways, we looked to what had been done previously in DMD to guide our clinical development. So, sometimes it's good to follow others that are blazing a trail in a specific disease because we learn a lot about that disease and how to develop a drug more optimally. And so a lot of what I'm going to share today on DMD was based on, others that tried to pave the way. What I'd like to talk about is how we take a technology that is very amenable to be easily adapted to different genetic subtypes in a disease like Duchenne, which is highly fragmented. And so as we continue to see the convergence of technology that can work at the genetic level in a very personalized genetic way and the understanding of the biology of disease, we're faced with challenges that we've never had to face before, and that is, how do you develop drugs in an era where you actually have a technology that can be applied to a variety of genetic subtypes in a disease, many of which are very rare and are infeasible to traditional drug development? And how do you approach this? And this is something that we all have struggled with, but we think we're getting closer to a pathway to do that, and that's what I'd like to talk about today. And this really came about because all these constituencies that are on this slide all are grappling with the same issue. Because it's one of these things where if we achieved success with this technology, then very quickly, right, every patient or parent of a boy with DMD is going to say, well, how can you apply that same technology to my son's genotype and that genetic defect? And so, patients are very acutely aware of this. The regulators are understanding that they don't want to be in between a technology that seems to be working and can be applied for these rare genetic subtypes and a parent who wants that technology for their child. Obviously, the advocacy organizations, the academic and government researchers who are involved in this space, they all understand this, and industry and the clinicians are also trying to figure out how do we do this. So what I'd like to talk about today is how we might apply this. This is just a slide on the type of technology that I'm talking about, and this is not a new technology. This is antisense, and antisense has been around for several decades now, and like many breakthrough technologies, we learn a lot along the way, and there's often a trail of failures before you figure out the best way to apply it. Our chemistry is called PMO, or morpholino, phosphorodiamidate morpholino oligomers, and we believe they're very differentiated from other RNA approaches based on the chemistry backbone. And so you can see how we've highlighted the way it works, the delivery, the stability, et cetera. We believe we have advantages across traditional RNA therapeutics. Now, you'll see the mechanism of action on traditional antisense has a plus and a minus, and that's because traditionally these other antisense chemistries were only applied for translation suppression or turning off gene translation, and now we're starting to see with companies like Prosensa, ISIS applying their technology in SMA, changing the mechanism to be more like our chemistry, which is steric hindrance, and being able to turn on gene translation and protein production. But I'd really like to talk about Duchenne and how we're trying to use a technology like this to try to make sure every patient can be treated who can benefit from this type of technology. And Claudia earlier talked about, the 13% of patients that could be treated with her mechanism. Well, the large majority of the rest of the patients in DMD can be treated with this technology that we call exon skipping. However, it's very fragmented. In fact, the largest group of patients that could be treated with any one drug is about the size of the Adalurin market, about 13% of the DMD patients. So what I'd like to describe today is an approach that we believe regulators may be amenable to how we could potentially get to every patient who could benefit from this technology being treated. Duchenne is a disease that's essentially caused by a lack of dystrophin. It's an X-linked chromosome disease that affects mostly boys, and in these boys, they have genetic defects that are out of frame and render them unable to see the read-through of the protein. So what we're essentially doing is trying to fix that defect so that we can turn an out-of-frame deletion into an in-frame deletion. And this idea is in practice in nature in the form of Becker muscular dystrophy, because Becker muscular dystrophy patients have in-frame deletions. They don't have a full dystrophin gene, and it's not a full-length protein, but yet this truncated protein is functional, and it produces a very different natural history, one that's much milder disease. In fact, many Becker patients live into their adult lives, sometimes old age, without even knowing they have a muscular dystrophy, or without ever requiring a wheelchair or attaining a loss of ambulation. So here's an example of how our technology works. This is a cross-section of the dystrophin gene. It's 79 exons long. And this is a region that is particularly ripe for mutations. And here's a patient. You can see they have a deletion, we refer to it as exons 49 and 50. That's considered out of frame. Okay, so you see the puzzle pieces don't fit together on this. And so, but we know what the bad actor is. In this case, if we can just silence exon 51 or hybridize that out of the reading frame, then we're able to have read-through and create an in-frame deletion. So we're converting a 49-50 deletion to a 49-50-51 deletion, which is a Becker genotype. And we think if we can do this, we might see the phenotypic changes, we might see the protein production, and we might see the phenotypic changes associated with that. And we have now proven that with our technology against exon 51. But here's the real dilemma, and I think this slide captures it well. You see the top 5 exons that if they were skipped, how many patients they could potentially treat. And many believe that you can really only feasibly do clinical safety and efficacy studies on the first one. That's the only one where you can find enough patients to power a study and get enough of a safety database to feel comfortable. You could argue that the next 2 or 3, maybe you could cobble together to look at safety and efficacy, or at least trends, maybe not powered sufficiently. But also we have competition. As previous presenter pointed out, we have a direct competitor that's also competing for patients. So that's another challenge that we're having to grapple with. But you can see after the first 5, it's a long tail. And none of those exons are amenable to traditional drug development and regulatory approval. And so this is the challenge that we're trying to meet, and we think we have a way to do this. So this is traditional drug development, okay? So there's a couple problems with this in DMD, right? So one is there's not enough patients. This is what we call an ultra-rare disease. And when you segment it by the genetic subtypes, there's even fewer patients. So the idea that you can do the traditional drug development to guide is infeasible. The other thing is this takes a long time. Now maybe a good biotech can shorten that span from 16 years here, may be a little bit overstated. But the idea is this is a disease where if we have to wait 10 years for a technology that seemingly is proven to be safe and efficacious for this disease, well, in 10 years, if you're a 10-year-old, right, you are going to lose all function of your arms and legs, maybe even pass away by that point, start to lose your cardiac function, your pulmonary function. 10 years from a 5-year-old, you're going to have lost all your ability to walk, probably on the way or already at ventilation. So 10 years is not amount of time that we have to wait once we have a proven technology. Now, this is particularly important when we have a technology that is already showing signs of efficacy and safety. Imagine if there's a technology that could be easily applied to all of these genetic subtypes, and the regulators said, sorry, you have to do this type of traditional drug development to get the next one approved. And we already talked about how it's infeasible because we don't even know if we could find the patients. So you can imagine it's a unacceptable answer to say, sorry, we have technology, but there's no way in the current guidelines to get it approved. And we know the other reasons is it's costly, right? The timelines are increasing. This is traditional drug development. And in these ultra-rare subtypes, we all know that companies do return on investment. Is it worth it to do all of these studies to treat a population that may be 1% of all DMD patients, right? Very quickly, you then would have businesses that say it's not worth it. We're only going to focus on the most prevalent. And I think all of us would agree, well, that's not fair, that someone lost the genetic lottery, and therefore we have technology, but we don't have a way to make that drug or technology available. And so, you know, in our exon skipping platform, this is the perfect example, right, where we have the technical feasibility, but not to apply it in the same way that we would do regulatory and clinical development in the traditional sense. And the good news is that I think all of the stakeholders in the DMD community get this, right? They understand that we can't wait that long. And there's organizations, there's many of them, some of them represented here today, like Parents Project Muscular Dystrophy. There's many small organizations, large organizations, but all of them would like to see good technology applied broadly for all the patients that could benefit. And there's many academic centers that we're working directly with who get grants to do additional research in DMD, and they recognize this. Eric Hoffman being one, we collaborate with him. He was an earlier presenter on how we can apply DMD learnings to other applications. And the FDA is also said many progressive things and are behaving in a way that's very new and fresh than how they've behaved in the area of rare disease. Now, that may be also because Congress sent a mandate out with PDUFA 5 and FDASIA that we really need the FDA to think differently about rare diseases and applying risk-benefit and how they think about approvals and accelerated approvals and the like in this disease area. And here's an example, right? These are three very senior members of the FDA hierarchy and the FDA commissioner herself who've highlighted, and this is very recent. This is, these are speeches that have been given this year. So you have Peggy Hamburg saying, FDA is trying to respond by achieving smarter regulation, greater flexibility, and creative approaches to drug development and evaluation. You have Bob Temple from Cedar saying, we think people are willing to accept risk when they have a terrible disease with no treatments, and this would describe DMD. You accept less data to provide a treatment where none exists. And Janet Woodcock, in a speech recently, said, we hope for seamless programs that take the least time possible to verify effects and gather data. We expect many drug candidates, she was speaking about breakthrough designation, would come available very quickly with, her words, phase 1 data. So this is sending a message that if you have a drug that is clearly having an effect and is safe or safer than what you would expect in a disease that's devastating and has morbidity and sequelae associated with it, like DMD, that they are flexible. So I'd like to describe the potential path to what we describe as a class approval. And we describe it as class approval because we would be using the same technology, the same backbone chemistry, and based on the learnings and the database that we create for the first drug, we leverage that to gather more data for the second, third drug, maybe more, that we can then say, okay, is this burden of proof here where we can connect the dots, where this in vitro screening of exon skipping produced at this standard dose a level of dystrophin of a certain range that is translating to an efficacious benefit clinically, and it's safe. Well, when you do that with the second drug, we show dystrophin based on in vitro data that shows exon skipping at a standard dose, and we follow these patients, you know, we could follow them post-marketing and show that they've stabilized. We do that with a third. At what point do we say, okay, that's enough? And of course, we would expect post-marketing commitments to manage safety, maybe have examples of dystrophin, maybe N of 1. Vertex earlier described N of 1 type situations in the cystic fibrosis area. So already the FDA is applying very different standards on, you know, these technologies where it's very fragmented and can be easily applied to genetic subtypes. And the idea is that once we have that, if we keep everything else constant, how we manufacture the drug, the dose that we're using, the in vitro assays in which we're measuring the efficacy in vitro, that you can start to have confidence in that standard and that consistency. So I'll go through these steps in a little more detail. So again, here's a backbone chemistry that we use for eteplirsen, is the name of our lead program for DMD. It's got very good benefits. It's, we know a lot about this chemistry. It's actually, this company was founded in 1980, and it was founded on a chemistry that was based on trying to get around the deficiencies of the phosphorothioate chemistry of some of the competitive chemistries in the RNA space. But we know a lot about this. We know a lot about the safety profile and even other diseases outside of DMD. And so it starts with understanding your drug and your drug chemistry and how it can be applied. Then obviously we've done a clinical study, a phase 2B study, where we looked at 2 doses, and we looked at biochemical response with dystrophin. We looked at clinical outcome measures over time, and we looked at safety. Again, we saw very robust dystrophin levels. We captured dystrophin-positive fibers using immunohistochemistry, which is, again, an approach that PTC has used, that our competitor Prosensa is using, to understand and looking at the actual muscle tissue and where is the dystrophin localizing, how many fibers, where you typically would see none in a Duchenne patient, how many of those muscle fibers light up with dystrophin. And we were surprised ourselves when after 48 weeks of dosing, we saw about half of the fibers in each muscle biopsy sample were lighting up with dystrophin. And if you look at the images, those images look closer to a normal muscle tissue than they do a DMD. And that was very encouraging to us. And we saw actually increased levels over continued dosing, and that was encouraging. And then we looked at clinical outcomes, and this was also encouraging. You saw some graphs on 6-minute walk tests. Well, we had a small sample size, and at the time, we didn't have enough drug to do a larger study. And so we were faced with the challenge of how do you show a treatment effect with such a small sample size? So we expressly selected a patient population that we would expect would be on a more rapid decline and loss of ambulation over a 24, 48-week time frame. And so we started a treatment arm and a placebo arm. And what we saw was the treated, now we had 2 patients who rapidly progressed shortly after enrollment. They were actually twins who had the same genotype and same phenotype. And they dropped significantly after the first 4 weeks before we had a chance for the drug to produce dystrophin and to work. So we've censored those 2 twins from this analysis. But basically, the treated patients remain stable even though we selected them based on criteria where we would have expected them to decline. But more importantly and more impressively, the placebo patients who started to decline, and I'll say that the shaded part is where we know the dystrophin starts to materialize in the muscle tissue and when we would expect a stabilization based on the dystrophin effects. Claudia earlier showed that 350 and above, they're relatively stable, but below 350, well, these placebo patients who started in the high 390s, by the time we had dystrophin produced, they were in the low 300s. These are the ones who should be falling off the cliff, so to speak. And yet, even that more advanced population, we were able to stabilize based on the dystrophin levels I described earlier. So one of the keys to this idea of class approval is establishing a surrogate, right, that we can have some faith and trust in, that we can extrapolate to these other genotypes. And we believe dystrophin is that measure in DMD. We know that disease exists because of lack of dystrophin, and we think restoring dystrophin is translating, in this case, to a clinical benefit from the drug. So what you see on the bottom here is something that largely has been described in the legislation, and the idea of more accelerated approvals is the idea you have a disease, it's very devastating. Is there a surrogate marker like dystrophin that would, as FDA verbiage states, would be reasonably likely to predict a clinical benefit? So once we can establish that, and we believe we've done this with our phase 2B study, We have 3 other exon skipping drugs right behind it that are in preclinical testing right now. We've identified lead sequences for each of these, and these are the next most prevalent exons. They're in the top 5 or so. And so this is where we're hopeful that once we get these drugs in the clinic and we can show the exon skipping efficiency would translate to a standard dose, and then we just need to test these drugs in some additional patients. And if we can show similar pharmacokinetics, similar safety profile, and at least the trend, or maybe even demonstrate the clinical benefit, then we start to put the links together. We start to see how A translates to B, translates to C, so that we can start to back off on that same burden of proof for the more rare indications. And of course, the only way that we think you can feasibly treat every DMD boy who could benefit from this technology is you need to standardize and you need to extrapolate. If we were to approach this dilemma with approaching every single sequence with, well, we need to do dose optimization, and maybe you need a 30 milligram dose with exon 51, but maybe it's only 25 milligram per kilograms per week with exon 53. Well, as soon as you go down that path, it becomes infeasible to ever imagine that you can get beyond the top 2 or 3. And we believe the regulators get this and understand it, right? And it's tough because researchers, right, they like to, dig deeper, find the differences, right, find the nuances, and how are things different rather than how are they the same. But we believe in DMD and this type of technology that there are more similarities than differences. That extrapolating, okay, maybe we're overdosing a little bit in this population and underdosing over here, but if it's producing a dystrophin level that is clinically meaningful, that's what we're trying to achieve. And it means the difference between, more than 50%, probably more than 70% of patients who could benefit from this technology get a treatment or not, based on how the regulators would approach this. And then, of course, everybody's like, well, gee, how can you approve a drug if it hasn't been tested before approval? Well, again, we're using a different burden of proof based on similar chemistry. All we're doing is taking the same, we call them subunits, the ACTs and Gs, and we're moving them around in a different order. Okay, a different sequence and a different length. It's the same drug material that's being used, and it's just being configured. And we already have evidence that when you dose this chemistry, the pharmacokinetics are essentially identical. It's cleared the same way. We're not seeing the safety problems. But again, there's no precedent, so we're really having to collaborate with the regulators to help us with this pathway. And of course, because of that, we would expect that they'd say, well, because we just don't know, we think you need to track the safety of these patients, even if it's a few, but make sure that you're capturing some safety data. And so, of course, we would do that. And maybe some longitudinal data to show, yeah, in fact, they are more stable than we would expect, natural history would show. And so, again, we believe this type of innovative drug development, and I would say innovative regulatory science, is the right pathway when you're dealing with a disease like Duchenne, where we have a technology that seemingly is working, is safe, and these boys have no other options, right? We know the effects of the disease. They're devastating and they're irreversible, and we don't believe, even with this technology, that once a boy is in a wheelchair, that he'll take the drug and be able to get up and walk again, because he's lost muscle mass. This is not going to reverse, right, Duchenne. And so we think time is of the essence, and we think it's appropriate that we try every way possible to make sure we can use this technology in even the rarest genetic mutations in Duchenne. And so we think it's the right thing to do. We think the regulators know that. We know the advocacy organizations know this. And it's a partnership that we believe, we hope that when we look back a few years from now, that we can say, we all banded together and we did the right thing to make sure we could get this technology to all the boys that could benefit. And then, of course, this technology is amenable to a lot of other diseases, a lot of other rare genetic diseases. So this is not a problem that is just confined to Duchenne muscular dystrophy. There are many rare genetic diseases and orphan indications that are going to require a different regulatory approach and a different way of assessing technology and risk-benefit. And so we think DMD can blaze the trail. This technology can blaze the trail so that all these other diseases that we are starting to understand the biology of and that we can easily adapt and apply this technology can really help a lot of other patients out there with other diseases. So thank you very much.